Welcome to one of the most fascinating and mind-bending topics in genetics. Today, we are diving into the fundamentals of genetics, specifically focusing on shell coiling in the freshwater snail Limnaea. We will explore a beautiful exception to standard Mendelian genetics called the maternal effect. By the end of this video, you will understand how an organism's physical traits can be entirely dictated by its mother's DNA, completely ignoring the genes it actually inherited. Let us get started. Before we look at snails, let us build some intuition. Imagine you are baking a cake, and you have a recipe that specifically calls for a square shape. However, the person who prepared the kitchen before you arrived only left you a round baking pan. No matter what your recipe says, the moment you pour the batter into that pan, your cake is going to be round. In genetics, we usually assume that your own personal recipe, your DNA, determines your physical traits. But sometimes your mother pre-packages your very first environment, the egg cell, so strongly that her genes determine your physical traits. This completely ignores the DNA you actually inherited. Now that we understand what a maternal effect is intuitively, let us look at its most famous classic example. In formal genetic terms, a maternal effect occurs when the phenotype of an organism is determined exclusively by the genotype of its mother, regardless of the organism's own genotype or the father's genotype. We see this perfectly in a species of freshwater snail known as Limnaea peregrina. The shell of this snail can coil in one of two directions, to the right, which is called dextral, or to the left, which is called sinistral. The direction of this coiling is controlled by a single gene with two alleles, where the dextral allele is completely dominant over the sinistral allele. This brings us to an important question. Why does the mother's DNA have such absolute, overpowering control over the baby snail's shell? The answer lies in the very first moments of embryonic development, long before the baby snail's own DNA is even activated. While the mother is forming her eggs, a process called oogenesis, her own diploid cells produce specific proteins and messenger RNAs based on her own genotype. She pumps these gene products directly into the cytoplasm of the unfertilized egg. She's essentially stocking the kitchen with her own tools before the baby's recipe is even opened. Following up on those maternal proteins, let us see what happens once the egg is fertilized. When the egg begins to divide, these maternal proteins act like microscopic scaffolding. They physically dictate the angle and orientation of the first cell divisions, known as the mitotic cleavage spindle. If the mother has at least one dominant allele, she provides right-handed scaffolding proteins, forcing the embryo's cells to spiral to the right. By the time the baby snail's own genes finally turn on, the physical spiral of its body is already permanently locked in place, making it impossible to change direction. Now that we understand the biological mechanism, let us represent this mathematically. To understand this logically, we must separate the concept of an individual's genotype from their physical phenotype. In standard genetics, an individual's phenotype is a direct function of their own genotype, but here we write a different relationship. The formula states that PO equals F of GM. PO represents the physical phenotype of the offspring, which is the actual direction the shell coils. GM represents the genetic genotype of the mother. The function F represents the biological process of the mother depositing proteins into the egg's cytoplasm. If the mother's genotype contains a dominant allele, the offspring will be dextral. even if the offspring itself is entirely recessive. If the mother is recessive, the offspring is sinistral. Before we move on, let us see this formula in action with a concrete example. Let us walk through a classic genetic cross to see how this plays out. Suppose we cross a pure-breeding dextral female snail with a pure-breeding sinistral male snail. We want to find the genotypes and phenotypes of the first generation. The mother is homozygous dominant and the father is homozygous recessive, so they pass on a dominant and a recessive allele respectively, making the F1 generation genotype heterozygous. Now, to find the F1 phenotype, we look at the mother's genotype, which is homozygous dominant. Because she has the dominant allele, she deposits dextral-forming proteins into her eggs. Therefore, 100% of the F1 offspring will physically have right-handed dextral shells. Now that we have our F1 generation, let us allow these snails to mate with each other to produce the F2 generation. When we cross the heterozygous F1 snails, standard Mendelian genetics gives us a genotypic ratio of 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Normally, this would result in a 3-to-1 phenotypic ratio. However, to find the F2 phenotype, we must look at the genotype of the F2's mother. The mother of the F2 generation is the F1 snail, whose genotype is heterozygous. Because the F1 mother has a dominant allele, she produces dextral-forming proteins for all her eggs. As a result, every single snail in the F2 generation will be dextral, regardless of their own genotype. The expected Mendelian ratio is completely masked. So, we have seen the standard cross. This brings us to the ultimate proof of the maternal effect, the reciprocal cross. Geneticists perform this by swapping the sexes of the parents from the previous example. This time, we cross a pure-breeding sinistral female snail with a pure-breeding dextral male snail. Just like before, the mother provides a recessive allele, and the father provides a dominant allele, making the F1 genotype heterozygous. But what is their phenotype? We must look at the mother's genotype, which is homozygous recessive. Because the mother lacks the dominant allele, she deposits sinistral-forming proteins into her eggs. Therefore, 100% of the F1 offspring will have sinistral shells. This is incredible because these F1 snails possess the dominant allele, yet they are physically left-handed. Now for the final mind-boggling step of our proof. Let us cross these physically sinistral F1 snails with each other to create the F2 generation. The F2 genotypes are again 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Now, pay very close attention to determine their phenotype. We must look at the F1 mother. The F1 mother is physically sinistral, but her genotype is heterozygous. The maternal effect rule states that the offspring's phenotype is determined by the mother's genotype, not her physical appearance. Because the F1 mother's genotype contains the dominant allele, she produces dextral-forming proteins. Therefore, 100% of the F2 offspring will be dextral. A physically left-handed mother just gave birth to an entire generation of right-handed babies, simply because her hidden DNA contained the dominant allele. Let us take a moment to look at this diagram, which beautifully summarizes everything we just proved. On the left, you see the standard cross, where a right-handed mother produces right-handed F1 offspring. On the right, you see the reciprocal cross, where a left-handed mother produces left-handed F1 offspring, even though both F1 generations have the exact same heterozygous genotype. Notice what happens when we move down to the F2 generation at the bottom. In both cases, the F2 generation is entirely right-handed. This visual perfectly captures the essence of the maternal effect. The physical spiral of the shell is always one generation behind the genetic code that actually determines it. We have covered a lot of ground today, so let us summarize the key takeaways. First, the maternal effect teaches us that inheritance is not always as simple as reading the DNA of the offspring. In Limnaea shell coiling, the mother's genotype dictates the physical environment of the egg cell, permanently locking in the offspring's physical traits before its own genes can intervene. Always remember the golden rule of this topic. To know what a snail looks like, you must ignore its own genes and look exclusively at the genotype of its mother. You have now mastered one of the most unique inheritance patterns in biology, bridging the gap between classical genetics and early embryonic development. Great job!